From the title of my talk, you already guessed that we are focused on chronic disease, and I'll explain why in the coming slides. So let's dive right in. This talk will be divided into two parts. First, I will introduce the links between chronic disease and metabolism, and why it's relevant to study this type of disease with metabolomics. And in the second part, I will present a couple of case studies where I will show you, especially using the MxP Quant 1000 kit, how to screen samples, leverage data, and translate insights. So we're going to look at an example of targeting the microbiome in the context of inflammation, and another one that will focus on energy metabolism in the context of Alzheimer's disease. But first, what are those links between chronic disease and metabolism? So the WHO, the World Health Organization, over 10 years ago issued a global action plan for the prevention and control of non-communicable disease or chronic disease by 2020. So as you can see on the slide, whether you look at diabetes, dementia, or certain forms of cancers, these actions did not manage to curb the tide. And by 2021, globally, 75% of non-pandemic related deaths were attributed to chronic or non-communicable disease. Now, a couple of years ago, we put together a white paper that you see here on the left, where we looked at the links between metabolism and chronic disease and how metabolomics can read this. And what we found out is that there are very deep links between those different diseases that you wouldn't necessarily bring together, like Alzheimer's, autoimmune disease, or type 2 diabetes, and that the root of those connections can be found in what we eat and the impact it has on our microbiome. So this is what you see here on the graphic. We all know this idea of the balance or imbalance of the gut microbiome. And when you look at this through the prism of metabolism, this results in different types of metabolites that are generated by our microbiome and that become signals for our bodies. And this has direct effects in the gut initially, and then in the immune system around the gut, and then it spreads to other organs as well. And so what you'll typically see in individuals who, especially over time, develop this kind of imbalance and sustain this type of imbalance is inflammation, which is an underlying cause of all the diseases that I will discuss, and cellular dysfunction, such as mitochondrial dysfunction, organ dysfunction, and eventually different types of disease. So this strong influence of our environment, which is diet and lifestyle, makes it crucial to study chronic disease with omic technologies to get this more granular measure of health. And metabolomics is an especially interesting omic in that regard because it is the one that is most sensitive to environmental changes. It's a omic that is going to fluctuate and respond, adapt to our environment, much more than, for example, our genome, which is fixed, and more sensitively also than other intermediary omics like epigenomics or transcriptomics or proteomics that are going to be sensitive to both genome and environment, but they're not going to be as sensitive to the impact of, for example, diet. So that's why we always encourage people, especially when they want to combine omics, to include metabolomics in this approach. It's very easily combined with genomics in MGWAS studies, for example, and it really brings this phenotypic aspect very quickly and adds very complementary information on health to other omics. So going back to our white paper, if you go to that document, you will see that we treated, or we studied 6 different diseases. These are cancer, autoimmune diseases, Alzheimer's disease, depression, and so the autoimmune diseases are IBD, multiple sclerosis, and type 1 diabetes. And going through the literature, we found we made a combination of their metabolomic profile either at the beginning or onset of the disease and also in later stages, and also the symptoms and what is known about the disease. And so we built models that are based on metabolism to try and understand what these diseases look like at the metabolic level and also how they come to be. And what we found really striking is that you could always link things back to certain metabolites, and especially certain microbial metabolites, such as short-chain fatty acids, bile acids, or indole derivatives, so metabolites of tryptophan. And you could always find links, especially in the onset or what could be the onset of the disease. Then, of course, we often found links to energy metabolism, nitrogen metabolism, which is more and more studied, metabolites related in immune regulation, and also lipids that can be strong signals and also markers of a disease that comes to be. And so this was a great inspiration for the MxP Quant 1000 panel. When you want to target analytes, you want to find the ones that are going to be most relevant for the study that you want to make. And so this white paper, where we saw this common ground of all these chronic diseases, was a huge inspiration to design the panel for the MxP Quant 1000. And why did we want to build such a panel? Is because we want to make it possible for metabolomics to become a driver for 5P medicine. Metabolomics is often seen as a source of biomarkers, and it is. It's a great source of biomarkers, but it can do much more than that. And when you look at medicine through the lens of 5P medicine, you look for preventive medicine, predictive, precision medicine, population-based medicine, and participatory medicine angles, and ways to use science and research to promote these relatively novel ways of doing medicine. So when you look at this from the perspective of metabolomics, for example, for preventive medicine, some of the uses you can make of metabolomics are, for example, calculating metabolic risk scores. You can also design and monitor interventions, whether they are therapeutic or lifestyle interventions, like dietary interventions. And metabolomics is going to be a very sensitive way to first detect which can be the best ways to intervene, but also monitor whether they're having the expected effect or not. For predictive medicine, biomarkers are the typical way that people know how to use metabolomics. Usually you have a well-designed study and a good machine learning algorithm, and you're going to find the most relevant biomarkers for your study. But there are many other ways to use metabolomics for predictive medicine, especially, for example, in drug design or in studying drug response. Precision medicine is probably the best known of these 5P. Here, typically, what you will do with metabolomics is you will stratify groups of patients. So you will have larger groups, and then you will find different profiles that allow to separate them into smaller groups that can be addressed individually or separately. And this stratification is also called metabotyping by people who are quite used to metabolomics. And this can lead not just to a better description of symptoms or of response to drugs, but also to personalized treatments. Another very important aspect in the use of metabolomics in medicine and the future uses of medicine is population-based medicine. So here is a typical example where metabolomics is used for the power that it gives to understand the effect of the environment on health. So you're going to use metabolomics to study the environmental variance as opposed to the genetic variance. And then you can describe subphenotypes and use that in multi-omics approaches, which you can use in other contexts, but is especially well liked in population-based research to understand the different subgroups of the population. And last but not least, participatory medicine is, I think, one of the aspects that is going to come up on top in the next few years. What we mean by participatory here is that people, first of all, can, for example, access medicine, and especially the more modern aspects of medicine, from their own home. You can collect blood or other sample types at home and send it in the mail to your doctor or to the clinic. And this has to be then compared to reference ranges in the same way that we compare measures of metabolites or other markers in blood to reference ranges in the broader population. And the idea here is that you can end up with patient-centric results. Today, this is starting to take hold in the mainstream way of doing medicine, but we're still at the very beginning. But one part of the world where this is quite used is what is called consumer health, where companies enable the general population to access, for example, their metabolomic profile. And I think this is something that eventually is going to merge with the more mainstream way of doing medicine. And here again, metabolomics can play a big role in giving access to this new measure of health. So as you see here, there are many ways to make use of metabolomics. And when you have a panel like the MxP Quant 1000 that is designed to better understand and target chronic disease, there's a lot that can be done combining these approaches. So let's look into some of the examples that we've put together. These case studies, and each time we're going to look at a three-tiered approach. First, screening the samples, analyzing them with the kit, and getting a broad metabolomics and lipidomics profile that we're going to be able to leverage then with computational approaches, and also, for example, by calculating sums and ratios of metabolites that are known to have relevance to biology and to medicine. And then from that knowledge, we can translate the insights and find the innovative solutions that are needed to answer the questions that you're trying to answer. So my first example has to do with the microbiome, and it's one I really like to put forward because I think more and more we understand that our gut microbiome and our microbiome in general, but our gut microbiome in particular, is very deeply linked to our health status, and it has a strong link, especially in the context of chronic disease. So here we have a panel of several derivatives of tryptophan, which is an essential amino acid that is typically metabolized in our gut by our microbiome through different pathways. And a balance between the indole derivatives and the kynurenine derivatives have been shown to be really important and a good reflection of the inflammatory status for the host. And so here you see in green some of the tryptophan derivatives that are quantified with the MxP Quant 1000 panel. And so depending on your study and your study design, you're going to have different groups that you want to compare, but you're going to quantify all of those metabolites in there. And if the metabolite is there above the limit of detection, is going to be first detected and then quantified with the kit. Now, the question is, if I know nothing about metabolism, what can I make of this? So after screening, you have this high quality data, but how do you make sense of it? This is where the leveraging step comes in. And within our software that comes, that works with the MxP Quant 1000, we have this module called Metabo Indicator. And there, automatically, for all the metabolites that could be quantified in the sample, sums and ratios, and only relevant sums and ratios are calculated. And you also get the literature references that give you access to a deeper understanding of what's happening there. So here, for example, what we could typically look at is the ratio of kynurenine to tryptophan. It's a very well-known ratio. It's even used in the clinic by some clinicians, and it's a known marker of inflammation. So when you detect that in the blood, if you see that increase in the blood of patients, you can assume that the immune system is at work activating inflammation. There are many other metabolic indicators that will be calculated from this subpanel that I showed you. But this is just one example where you can start to think, okay, what could be the reasons why this is increased, and does that match other parts of the panel that are also related to inflammation, for example? And then based on that, and also still in our context of 5P medicine, which I really like to use as a kind of framework to understand disease and to look for solutions to the medical problems we have today, you can think of many different types of solutions that can be built from this information. If you're looking for preventive measures, for example, intervention is of course something that we intuitively know can be a good way to influence our microbiome. And there are the three typical ways of addressing this with prebiotics, probiotics, and postbiotics. So a prebiotic approach, when you have this imbalance between indole and kynurenine derivatives, could be to change the food that you provide to your microbiome. So, for example, having a high fiber diet. For probiotics, you want to directly modulate which microbial strains are in the gut. So you ingest the bacteria directly. So you can give different types of bacteria that are going to promote one of the pathways that you're looking for. And for postbiotic approaches, this is where you directly supplement the metabolites, so the effector of these changes in microbiome. For example, indole derivatives that are going to have a positive effect on the immune system in the gut. But you may also be interested in predictive solutions, for example, predicting drug response in clinical trials. Here, there's a very interesting paper that came out a couple of years ago that showed using a broad screening, which you see here on the volcano plot, so broad screening of the serum of patients in a pancreatic cancer study, and just looking at the differences between people who will or will not respond to treatment. For example, here, 3-indole acetic acid, or 3-IAA, was a big discriminant between the two groups, and that was leveraged then by the scientists in that study to better understand the mechanism, but also to think of ways to increase drug response. And for example, in precision medicine, you can look for targeted chemosensitizers. So molecules, in that case a supplement with 3-IAA, that are going to increase the response to treatment for the responders, or also just turn non-responders into responders. So that approach worked really well in the mouse model in this study, and it's going to have to be then tested in humans in the next step. So this gives you, I think, a really nice overview of the different angles that can be used from the exact same information originally, but based on what you actually need for your research to leverage metabolomics to really find the answers to your scientific questions. So if you're interested in the microbiome and want to learn more about how to use the MxP Quant 1000 kit in microbiome research, I really encourage you to go to our application note. You can find it on our website. It's called MxP Quant 1000 in microbiome research, and it gives lots of examples with different chronic illnesses like IBD, cardiometabolic disease, cancer, and neurology, where you see applications of how scientists have leverage metabolomics and understanding of metabolism to combine microbiome research and the research in the disease they were interested in. Now, my second example has to do with Alzheimer's disease, so a much more specific disease than inflammation, and it's linked to energy metabolism. I call this energy crisis here because it's well known that in Alzheimer's disease, glucose metabolism is compromised in the brain, especially in neurons. And so when we do a broad screen in the blood of different metabolites, you see here in green again the ones that are measured with the MxP Quant 1000 kit. So you have a nice coverage of the TCA cycle and also of lots of analytes that come and feed into it. So you're going to get your picture first, your quantitative picture of metabolism, and then you can start to think of how that relates to the disease you're interested in this case, Alzheimer's disease. But let's say you don't have so much prior knowledge. The easy way to leverage these insights is by using, again, metabo indicator. Here, for example, we have an indicator that's a ratio of TCA cycle intermediates to amino acids that feed into that cycle. And then when you see an imbalance in this or a difference between your groups, for this indicator, you can start to think, okay, maybe there's a compensation of a lack of typical energy production through TCA cycle by a use of amino acids. And this is something that's often seen in diabetes, is a reduction in circulating amino acids, which could very well link with a compensating mechanism for energy metabolism. How do you leverage this? Again, different ways depending on the application you're looking for. Of course, for predictive medicine, you can be looking, and we're still looking for them, for prodromal markers of Alzheimer's, so before onset of the disease. So for this, it's really interesting to find markers that relate to what we know the disease will look like later on, and maybe early compensating mechanisms. Of course, you can also try to prevent based on that knowledge using supplementation. And for precision medicine, You can think of individualized, personalized intervention with dietary plans, for example, to try and very early on, let's say, reestablish the balance or promote energy metabolism to sustain healthy brain function for as long as possible. So here in a few minutes, I showed you these two examples of how we can understand disease better with metabolomics, but I hope that this gives you a sense that you too can start to transform medicine with metabolomics. So we've seen here a couple of examples where we can really easily screen the biology using this targeted panel of the MxP Quant 1000 kit. Again, the analytes were handpicked and chosen because of their relevance to many types of chronic disease. So it's really a broad, relevant panel to use in that context. Then you can use MetaboIndicator and other computational methods to leverage that knowledge. The advantage of MetaboIndicator is that it helps you to very quickly link that to relevant literature, because the sums and ratios that are calculated are based on existing literature that has been relevant in different biomedical contexts. And then depending on the application you're looking for, you can translate that into the innovative solutions that you need. So I hope that in these few minutes I could pique your interest in terms of the applications that are possible for this kit.